So the next speaker is from Oxford University in Spain. He'll be talking about functional imaging of psilocybin in rat.
Hello, it's very good to be here today. I'm very glad to have the opportunity to present some of this work here to some interested parties. So I'm going to tell you about some of the work that we've done with functional imaging, look at the functional imaging effects of psilocybin administration in the rat, and how we've looked at some of the specific underlying hemodynamic and neuronal factors that influence the functional imaging signals. So I'm sure you're all aware of this, so I'm just going to go over it a little bit to recap some of the most important bits. We used a drug called psilocybin, which is actually an active metabolite of psilocybin, and it's thought to account for the vast majority of the activity of... Sorry. I'm sorry, so psilocybin is the active metabolite of psilocybin, is that better? And it's thought to account for the majority of psilocybin's effects in vivo. Psilocybin is a serotonin receptor agonist, as we've heard several times today, and like other classic hallucinogens, it acts on the 5-HT2A receptor, but it's actually quite a dirty drug, and the 2A receptor is not even the drug that it has the greatest affinity for. It has similar or higher levels of affinity to the 5-HT1D, 2B, 2C receptors. And this is significant because of the wide distribution of serotonin receptors throughout the CNS, and also across different cell types. So the serotonin receptors are not just located on neurons. You find them on the ependymal cells in the choroid plexus, or on vascular smooth muscle on blood vessel walls, which is quite important, as we'll see, for functional imaging. So we'll just briefly blast through some of the functional imaging work in humans. So as we've seen in Robin's work this morning, they find exclusively decreased BOLD and CBF responses in humans to psilocybin administration. They also find that psilocybin alters functional connectivity. And we tend to see he observes his decreases in flow of BOLD signal in areas of sort of prefrontal cortex related to executive control. Now this stands in contrast to some PET studies which show that there's increased glucose metabolism in those same areas. That doesn't really make any sense superficially, because in an area where you've got increased glucose metabolism, you'd really expect to see increased BOLD signal or increased CBF. And this is the kind of question that we hope that we can address with animal studies. Because when we use our animals, we can use some invasive techniques that really aren't available to us in humans. So we can implant electrodes, or we can stimulate and manipulate pathways that are very well characterized and see what effects the drugs are having on those. And we can invasively measure blood flow. And ultimately, what we can do with that kind of information is, if we're interested in developing a hallucinogen as a depression treatment or an OCD treatment, we can understand more about how the drug works, develop the imaging as a biomarker for these drugs, or maybe even understand a little bit more about the disorders that we want to treat by knowing what's effective in treatment. My primary interest is in understanding the BOLD signal, however, and that's what I'm going to come back to here, and I think it's really important. I can't stress enough that the BOLD signal is a massively indirect measure of neural activity. We've got all these different factors that feed in, like deoxyhemoglobin concentration, anatomical differences in neurovascular coupling, and modulatory neurotransmitters. And these are all interacting in some unknown sort of way. And they give us these changes in deoxyhemoglobin signal that we see that give us the BOLD response. When we see those nice activation maps, we're looking at changes in deoxyhemoglobin concentration. And the reason that we have come to do some of this psilocybin work is that what we're interested in is these modulatory neurotransmitters. And these are kind of questions that we can really probe with using drugs, and they're a good example of why it can be very difficult to interpret BOLD and functional imaging signals. So for instance, with serotonin, primary serotonin nucleus in the brain is the dorsal raphe nucleus, which is partial inhibitory control of the locus coeruleus. And just a couple of weeks ago, Ethan Hamel's lab had a paper out showing that locus coeruleus activation alters the cerebral blood flow response that we see in response to stimulating a rat's whiskers. Not only that, but as I mentioned earlier, we have serotonin receptors located on blood vessel walls. So we know that because we use blood serotonin drugs that cause blood vessel constrictions to treat migraine. So this is just one of these many factors that can be altered in any way, it can influence cerebral blood flow itself and create great difficulties interpreting bold. And we're starting to understand more and more about the bold signal, but our understanding of it really is not complete. And all these problems are all multiplied manifold when it comes to negative bold. And we have a massive problem with interpreting about negative bold in that we really don't understand what it means. And there have been lots of theories proposed over time. There's the now discredited idea that it was vascular steel. Perhaps a few more people think now that it's related to fire inhibitory GABAergic neurons, which use deoxyhemoglobin without increasing blood flow. But this is something else that we can understand, and we can use drug manipulations to carry out this kind of study. So, to start off, we put our rats in the scanner because that was the obvious thing to do to see what happened to functional imaging signals in the rat. So, unlike Caroline and Flavy's rats, our animals are anaesthetized, and we tracheotomize them and ventilate them, and this is in order to keep various physiological and respiratory parameters that might confound the bold signal under control. We put them in our magnet at 9.4 Tesla and scan them for 15 minutes and then put in the drug and continue scanning for another 45 minutes. We use 1 or 2 doses of psilocybin and then we analyse our data in FSL. And unlike some of the other MRI work we've seen, we've not looked at default mode connectivity, we've not done that type of analysis. We've simply modelled that when the drug goes in, the bold response will either go up or down. That's all we've looked for, is just changes in bold response in any direction. So at this low dose, so we've selected this low dose as being equivalent to what Robin had used in his human studies. And we don't see any changes in either direction in our bold signal. Now, this may just be that it doesn't produce drugs, that we didn't scale the dose right, or that the anesthesia is dampening the effects of psilocin, which is a general feature of anesthesia, and it's another confound in bold interpretation in animal studies. But when we moved into this higher dose, we start to see both increases and decreases in bold signal in a variety of areas. So we see increased signal in subcortical areas, in the amygdala, in olfactory areas, and those kind of associated limbic areas in rats. Olfaction and limbic areas are very closely related. And the hypothalamus. We see decreased bold signal in various cortical areas, including the cingulate cortex, barrel cortex, entorhinal cortex, and parts of the hippocampus. And we see changes in both directions in the visual system, which was, well, it was nice to see changes in the visual system as a sanity check that hallucinogen is doing something that you expect it to do. So obviously the direction of our changes conflicts directly with what Robin sees, but we do see some anatomical overlap. So for instance, our effects on cingulate cortex and the hypothalamus and visual areas, these agree with the human studies. And for us, I mean, we thought that then, despite the differences in directionality of changes, we thought that it was still worth persisting with trying to investigate these bold responses a little bit further. And because we had changes in the barrel cortex, we thought maybe we can look at something that we know how to respond to as stimulation. So the barrel cortex is where sensory input from the whiskers is processed, and we can quite easily stimulate whiskers and have a look at the barrel cortex to see what's happening. So how we did this was we took our tracheotomized and anesthetized rats, and instead of putting them in a magnet, we put them under a laser speckle camera, and we get a little video like this out. So more red is more blood flow. So this is blood flow normally, then the stimulation comes on, and we see an increase in stimulation. And the blood flow response follows its own sort of fairly slow time course that develops after the end of the stimulations. As well as imaging the blood flow response, we were able to put in an electrode into that same area to record local field potentials. So local field potentials are a sort of mass measure of neuronal activity in an area. They're considered to be the best neuronal driver of the bold response. And we also inserted electrodes into the whisker pads to carry out stimulations. And another manipulation that we were able to use is to deliver carbon dioxide into the inhalation mix that the rat is breathing. And the reason that we did this is because it's a way of manipulating blood flow without manipulating neurons, so it's a nice control condition. Administering inhalated CO2 causes a massive and rapid increase in cerebral blood. And we did all these manipulations both before and after either the psilocin or the beam. So, with our stimulation results, what we see is a potentiation of the cerebral blood flow response to psilocin, to the whisker stimulation. So you can see here the red is the post-drug response to psilocin. But we don't see a change in the cerebral blood flow response to the CO2 challenge, which indicates that it's probably the changes in the response to the whisker stimulation aren't due to changes in vascular reserve or reactivity. Perhaps they have a neuronal source. So what do the neuronal results look like? Well, somewhat unexpectedly, the neuronal results show, we've not looked at power spectra like some other people here today have. We've just looked at the shape of the LFP response to stimulation. And we looked at the size of the response appears to be diminished, and it is also offset and delayed by a couple of milliseconds, as you can see. This is the red line here. So this again, it seems somewhat counterintuitive. You might not necessarily expect to see increased blood flow responses to a decreased neuronal response, but it does suggest that at least in the barrel cortex that we're seeing some alterations in neurovascular coupling brought about by psilocin. And this really has implications, I think, for interpretation of both studies using drugs like this, that in some areas at least we may be seeing neurovascular coupling effects as well as neuronal or even vascular effects. And we've also got a situation where we see a negative bulb response, but also we see a potentiated CBF response in the barrel cortex, which suggests that perhaps what the drug is doing is lowering baseline CBF, which gives the blood flow response more time more sort of room to increase in response to stimulation. And because of how we've collected our blood flow data, we're not able to actually tell that from our data. And I'm sort of here to urge caution in interpreting both findings because it's massively complex, and especially when we put drugs in, it makes the situation even worse. And finally, I'd like to thank the Wellcome Trust who fund my work, and my PI, Chris Martin, at Sheffield, who's at the Royal Society Fellow, and at Oxford, Trevor Sharp, Claire Howard, and the Citizen Bar.
The obvious question is, do we have to be doubtful about the very interesting results we had about this morning on the basis of Europe's?
I don't think so. I don't think necessarily. I was very wary of saying something like that when I came here today. I don't think that rather than David have over-extrapolated or over-interpreted their results really in any way. And I think that I was really interested to see their MEG studies, and thought that's really interesting because I had been worried that I was going to come in and contradict some of their work, but actually I think that we agree quite nicely with some of their MEG studies. It's really an illustration. This is one specific example in one part of the brain, and we know the coupling is different in all sorts of different bits of the brain, so it's sort of too isolated an example to say anything like that.
Can I ask you have?
I was just wondering whether the increases in the amygdala might have something to do with anxiety or stress in the rats that are anaesthetised.
Well, we anaesthetise them in order to reduce their anxiety and stress, so I hope not. Yeah, that's the purpose of the anaesthesia. We do see, since we're sort of put in, I'm going to speculate a little wildly here, but since we're putting in something that is going to stimulate sensory regions, and the amygdala is very close, the amygdala is almost part of the olfactory system in the rat, and we see lots of activation, we see lots of changes throughout the olfactory system in the rat. I would imagine that it's part of that network changing.
Maybe, yeah.
Yeah.
We also did the breath hold paradigm in the bowl study.
Ah, yes.
Actually, intentionally, I try not to make much of the neural change in the bowl signal, and I'm more interested in functional connectivity results and make stuff really in terms of what it's telling us about how the drug works in the brain, changes consciousness, etc.
Yeah, I remember that. I think I must have asked you before. You've done a breath hold.
Sorry, yes, we did this. So we did the breath hold, and we didn't find any significant
Yeah,
changes.
because breath hold is slightly different, I think, to hypercapnia. It's a little more neutral. And what we usually use in humans because it can be a bit distressing to people.
Yeah, it's open, of course.
Yes.
Another question could be related to the ASL versus bold. So this morning, you, Robin, presented both bold and ASL with overlapping results. And is it actually possible to do ASL as well in the rodents?
Yes, as of this week, my lab can do ASL in rodents. It's possible to do it. I'm sure other labs can do it. We just weren't able to do it at the time we were doing the study.
And now, since you presented these cautions about understanding the bold signal, and probably a lot of the audience is not don't come from imaging backgrounds. Maybe I don't know if you or Robin would maybe highlight the main differences between ASL and bold. That might be interesting for people now that we are going into this about understanding the bold. Just what is ASL providing?
I can tell you, if you like, ASL is providing an absolute quantitative measure of actual blood flow, whereas bold measures a change in hemoglobin, a change in the oxygenation state of hemoglobin, and that is primarily driven by changes in blood flow, but it's slightly different, and it's also it is also a relative measure. We can't quantify it, so we can quantify ASL, but not bold.
Given the discrepancy that you find between the effects of psilocybin in animals and humans, of course the translational aspect is an issue.
Yeah.
It always is. And I was wondering what do we know about the drug-induced effects on bold responses in animals relative to the ones that you see in humans? Can they be translated? Is there any information on that for drugs in general or for psilocybin specifically?
I think. So the information is limited, and this is something, because a lot of the work that we're planning to do is centered around drug manipulations of neurotransmitter systems. It's really an issue. I think the majority of the work has been done on manipulation of the dopamine system. And there's reasonably good agreement with the effects of dopaminergic drugs and on bold signals in animals and in humans. But the big confound is always anesthesia in animals. So, I mean, one of our next things will be getting away animals, but it can be done with in the past, using worms. Because obviously you have, interactions with anesthesias and dampening effects. I mean, that's the biggest barrier, to my mind, to translating from preclinical work to animals. And then, of course, you've anatomical differences, so this becomes an issue when you're looking at things like consciousness and very sort of higher order mental states, is that humans have a much better developed prefrontal cortex than rats. Rats have very little singular cortex or prefrontal cortex on the other side. So I guess you also have to have caveats related to what kind of drugs you're looking at and what sort of bits of the brain you expect to be activated, because they may be more or less relevant